Hello again, Biology 300 students. Mr. Parker here, and we are taking a look at the Genetics Session 2 screencast on classical genetics. There'll be a couple other here to follow on genetics, dealing with some of Mendel's laws and some of Mendel's patterns of inheritance. But this one we're going to talk about basically Gregor Mendel's his work and basically some of his conclusions that he came about with his experiments and some of his data collection that he had during his experiments. So moving on to here, some of the learning targets, as you can see, that we're going to kind of discuss in this particular screencast. In text pages, we're going to be looking at 8.1 and 8.2, chapters 8.1 and 8.2. And in your packet, you should have out page 59 and 69. And don't forget to have your screencast notes out, your template that you should be filling in the details of the main ideas as we go along through this. Okay. So moving on, here's Gregor Mendel. Gregor Mendel, he's the father, what they term as the father of genetics. He was a monk in Austria. 1865 was the time of his work, and what he was doing was studying the patterns of inheritance in peas. Now there was another scientist by the name of T.A. Knight who was also doing some experiments, kind of coming up with a lot of the same ideas, but basically what Mendel did was he took it one step further, and he really analyzed his data and looked at it a little bit closer. He went in and actually counted all of his results, and so basically you can say his detail was better, his data was better, because he went in and kept track of it all, and the numbers that came out were a bit, they had carried more weight because of the actual numbers he was using. They were building off his conclusions that he was making. Okay. So that, again, the gentleman's name was T.A. Knight, that was kind of going along with the same type of experiments that Mendel was looking at. Okay, so Mendel, as I mentioned, Mendel he chose pea plants, and the question is, the question here is stated is why did he choose pea plants? Okay, there's several different reasons why that he did. Okay, moving on to the next slide here will show us one reason. Okay, as it says here. The first reason is it's a self-fertilizing plant, which means it has the ability to produce its own by itself. As you can see in this diagram here, okay, you can see it has its male and female parts. It's able to reproduce, and Mendel made it a lot easier for him to work with because he could control this. If he wanted to let them self-pollinate, he could do that, or if he wanted them to, to manipulate them, he could cut off, he can cut the male parts here, the gametes, and then he can control what plants are fertilizing what plants, and see if he can get the results that he really wanted to achieve. So the first idea was that they were able to self-breed, self-fertilize, which meant they were true breeding. There was no crossbreeding between certain plants. Okay, and then moving on, looking at this little picture here. Okay. And what you can see in this diagram that's happening here, you can see Mendel is manipulating the plant here. Okay, you can see these are all white, okay, plants versus all purple. Purple in this case is the dominant trait that we're looking at. So we'll kind of talk about dominance here in a minute. But what he was doing was he was removing the male parts, okay, as it tells you here, and he was fertilizing the purple flower with the white flower. Okay. And what he ended up with, this is what we call the parental generation, or the P generation. And what he was ending up here with, in the first new generation that was coming, that they were all purple flowers. So he noticed that the whites, okay, the white plant or the flower disappeared. And we'll get into that a little bit more here in a second. But back to the idea of why he chose the pea plants. Again, they're self-fertilizing. That was one of them. Okay, there's many different forms that he had. And let's go ahead and take a look at what I mean by forms. Okay, so here, looking at this, you can see he's looking at round versus wrinkled, yellow, green, purple, white, inflated, pinched, long, short. So all these different forms you can look at and see if the numbers that he gets play out the same no matter what characteristic or trait he was looking at. Okay, so that was another one, the different forms. The plants were small typically. They grow easily and quickly. Okay, and that they produce many offspring. Okay, so these were the reasons why he wanted to use the pea plant. It gave him a lot to work with, a lot of resources, a lot of different traits to look at. He was able to manipulate and control some of the fertilization process in order to do that. Okay. So, looking at Mendel's work a little bit more closely here, what he again did, okay, up top you can see here, he was crossing at the P generation what they refer to as the true bred parents, meaning there was no cross between other organisms. They were basically bred with themselves. You have the purple here, okay, parent, which is the dominant trait in this particular case. In this case, the white flower is the recessive trait. And what he noticed at the P generation here, okay, he noticed the first generation, he noticed that all he got was the white, the purple, okay. So if we look at this here, let me go ahead and see if I can write on here for you. So what he came up with is a 4:0 ratio when we're talking about purple versus white. So purple is the dominant trait here. And then what he did here was he allowed these plants, okay, the purple plants, to self-pollinate themselves, and he ended up with this 3:1 ratio. So what he noticed was that this the purple flower, okay, was the dominant trait. Recessive white at the F1 generation was disappearing, and then it reappeared at the F2 generation, okay, in the numbers as you saw. So he used these terms here, as you can see, dominant versus recessive. Again, in this particular case, the purple was the dominant trait, and the white being the recessive trait. Okay. What I'd like to do is jump into one of our sheets. Okay. Let's go ahead and do that real quick. You can go ahead and flip to page 59 in your packet. We're going to take a look at this diagram real quick. And this is Mendel's famous experiment on looking at the purple and white flowers. Okay, so what you notice here and what you can see here, he allowed these plants, these initial plants, to self-pollinate. So in the P generation, what these are kind of referred to would be your homozygous. I'm going to throw some terms out there. We'll talk about them in a minute. Homozygous dominant, meaning that they're all dominant, bred with themselves, versus the homozygous recessive. Okay, so he allowed them to crossbreed, and by them crossbreeding, and the F1 generation, as you can see here, the first generation that came from that was the P generation. You notice here that he came up with this 4:0 ratio and all purple. And you should notice now that the white has disappeared. And this kind of surprised Mendel because originally he thought, he would get like a blending effect that occurred. But then what he decided to do is, all right, let's go ahead and have these self-pollinate themselves. Okay, and then what he noticed at the end, if you look at the numbers here on the bottom, he noticed that there is this 3:1 ratio. So the white reappeared. Okay, so this dominant traits, the F1 generation took over, the F2 generation, the recessive traits came back. And this was true for all the different types of forms that he looked at, no matter which flower color, seed shape, flower, where the flowers were located, that kind of stuff. Okay, so these were the numbers that he found very consistent with his experiment, all his experiments he was running through his lab. All right, let's just jump back in here real quick, go and do some more of our notes. Moving on from here a little bit about his experiments. So we're going to kind of get into a little bit more of these terms here in a second. All right. So a little bit more about Mendel's work. So what Mendel figured out was that these traits were passed on by what he referred to as factors. Okay. Now we know the factors now are called genes, but at the time Mendel was doing his work, they were just known as factors. And he knew these factors had more than one form. Okay. And what they were called were alleles. And there are at least 2 alleles for each trait. Now, there could be more than 2, but there's at least 2. So we're looking at this particular case here. We're looking at this chromosome, all right? And what we're looking at is flower color. And notice there's one here, and then we have another flower color here, okay? So those are what we refer to as alleles. They're different forms of the same gene, okay? So we're looking at the allele. There's the definition you kind of want to jot down. It's just a different form. It's underlined there for you. And we're going to move on to the next slide here. Okay, so now we're getting into a little bit more of the specifics on how Mendel kind of put his work together. So these alleles are represented by the first letter of the dominant traits. Okay, so make sure you jot that down. I would write that into your notes, that the alleles, okay, are represented by the first letter of the dominant trait. Okay, so for example, we've been looking at the pea plant and the flower color, looking at purple versus white, okay, and we know that purple is dominant over the recessive trait, so what we would have is big P is purple and little p is white. All right, and let's jump on into the next slide. Okay, so here you can see that now we're going to just give you a little quick little practice. So round seeds are dominant over wrinkled seeds. And you can kind of see on the bottom here, you can kind of see the answer. Big R equals round, the little r equals wrinkled. You have to use those letters. Okay, you have to use the dominant trait as the capital letter, and the lowercase letter is the recessive trait. Yellow seeds are dominant to green seeds, so yellow would be the dominant trait, and then little y would be green. And then tall versus short. Tall is the dominant trait. Short plants over short plants. So your big T equals tall, and little t equals the short. Okay. So moving forward from there, and then what he decided, what he needed to do was he was looking at describing the genetic makeup and also the physical appearance. Okay. So what we're going to look at here in this particular case, we're looking at tall versus short plants. Now remember, tall is the dominant trait, and short is the recessive. Okay, so your genotype is the actual genetic makeup of the organism, or what we refer to as the genes. I always kind of think of it as the letters. Okay, we're talking about it. So in this case, we could have a couple different combinations. It could be big T, it could be big T, or it could be little t, and we'll get into those here in a minute. But those are the different possibilities of combination when you're looking at this particular trait for size. Okay, and then the second one here is phenotype, and phenotype is the physical appearance or form that's observed. Okay, or I always think of physical phenotype kind of going together, and what we're talking about basically, this is kind of what does it look like. Okay, so in our case, this would be, is it tall or is it short? Okay, in this particular example, those are the two different physical appearances that we're looking at. Now we'll get into possibilities of having more than just two, but not particularly in this screencast. All right, so moving on to the next slide here. Okay, so now we're going to take a look. Now we know the letters. Okay, we know the capital letter gives us the dominant trait, lowercase gives us the recessive. We know how to determine that. We know what pheno and genotypes are. So now let's take a look at two other terms. This homologous, or basically homozygous, and heterozygous. Okay, we know that if you look at the prefix here, homo means the same. Okay, and on the bottom here, you know that hetero means different. So back to the top, the two alleles for a trait are identical. Okay, so you have two options here. You have big T, or little t. Now, your big T's are homozygous dominant, or also referred to as purebred dominant, and then you have homozygous recessive, also referred to as purebred recessive. Okay, so you should jot these down so you have an example in your notes on these. Then we look at heterozygous, and it's also referred to as a hybrid, where you have a big T, little t, versus the idea of having either all dominant or all recessive. Okay, and let's move us to the next slide. Okay, so we're looking again a little bit more at Mendel's work, and kind of summarizing this up, and this will kind of get us towards the end of this particular screencast. All right. And this is what we're looking at here, again, is Mendel's cross. He was going through the idea of, here's your purebreds. Okay, we now know truebreds as being like your homozygous, right? And what he found out was when he crossed these, okay, so he had the homozygous dominant, homozygous recessive cross, and he always came up with this 4:0 ratio in the middle. Okay, and then at the F2 generation, as it's stated here in the bottom, you always get this 3:1 ratio when he came about. Now, when we look at the genotype versus the phenotype on this slide, okay, what you should notice is when he did this cross of this F1 getting his F2 generation, when he crossed these those hybrids, or also referred to as heterozygotes, okay, he was crossing, in this case, it would be big P, little p, and big P, little p. And when he did that cross, he always got one big P, big P. He always got two heterozygotes, and you always got one homozygous recessive. All right, and so these are the genotypes. Okay, so if we were writing the ratio out, as you can see in the bottom here, the 1, 2, 1, you would have the genotypes would be big P, big P, little p, and two little p's. And the ratio would always be 1, 2 to 1 when you're looking at the numbers here. Now, when we look at the phenotype, we're looking at purple versus white. And what he found was when you make those, because remember what happens is this dominant trait is hiding the recessive trait. Okay, so what we have is 3 purple to 1 white. So whenever you cross two hybrids or two heterozygotes, you always came up with the numbers of a genotype of 1 to 1 and the phenotype of 3 to 1. Okay, and what I'm going to do now is jump to our next overhead real quick to kind of summarize things. And looking at this slide, this overhead, okay, this is page 69 in your packet. Okay, and let's go ahead and jot down some information real quick on this. So this is kind of reinforcing the idea that each parent has 2 separate factors or genes. We know that these separate forms are called alleles. Okay, so you just jot that down. Those separate forms are called alleles. When we're looking at this, the 2 parents, these cells right now are diploid. Okay, bringing back some old terms here. Diploid, both parents are diploid. We go through this process of meiosis because what we're trying to do is get into our individual sex cells. We know that the result of that is that all of these cells are going to be haploid. And when you're looking here, this parent is homozygous dominant. Okay, homozygous dominant. And this is homozygous recessive. Okay, and we're crossing those. So that's basically what you look at. This is our P generation if you're talking about Mendel's cross. So what he came up with here by crossing those, he did his fertilization process, and we know a fertilized egg is called a zygote. Okay, and what he always came up with here was this hybrid or heterozygote. He always came up with big Y, little y, and we know that as going to be a hetero, okay, or a zygote, okay, or a hybrid. Okay, and if you're looking at the numbers playing out, okay, remember what Mendel came up with. His genotypes were a 1-to-1 ratio if he was going to cross these heterozygotes now. So this was his F1 generation. Now if he moved on to his F2 generation, that's when he came up the ratio of the 1-to-1, okay, and then also the 3-to-1 purple to white. Okay, what I'm going to do is kind of wrap this up with a 3-minute video on Mendel, and then I'll come back real quick and summarize a few things, and then we'll be done with it. All right, with the screencast. Jump in. Here we go.
In the middle of the 19th century, an Augustinian monk named Gregor Mendel took up the question of biological inheritance with a series of experiments. Mendel had a naturally inquisitive mind and a profound love of nature. His scientific interests ranged from research on plants to meteorology and the theories of evolution. Working at a monastery in what is now the Czech Republic, Mendel started by crossbreeding different strains of garden peas, then observing the characteristics of their offspring. Why choose peas? He did it, he said, for the fun of the thing. Mendel noticed that when he crossed a round pea seed with a wrinkled one, the offspring were round, not a mix of the 2 characteristics as he had expected. Yet when he bred the round pea offspring, that's where the mix appeared, and the second generation had both round and wrinkled seeds. He continued to experiment, trying to understand what kind of biological mechanism would cause certain characteristics to disappear in the first generation, only to reappear in the second. Then one day, Mendel counted the number of peas in the second generation that had the wrinkled characteristic. Exactly 1/4 of the peas were wrinkled. What Mendel observed in his experiments were the biological phenomena we now refer to as dominance and segregation, only Mendel didn't know it yet. Still, his experiments produced a curious set of facts which, as he said, forced themselves upon my notice. You see, no matter how he crossbred the various strains of peas, the hidden characteristic showed up, but only in 1/4 of the second generation. For Mendel, here was the breakthrough. For the first time, he could demonstrate that the traits of successive generations were inherited in certain numerical ratios. In other words, there were fixed laws of nature that governed heredity. With this insight, Mendel made the first great discovery in the science of genetics. Each inherited characteristic must be decided by a pair of what he called factors. Each parent, he said, contributes one factor for each characteristic. Certain factors are dominant and others recessive, depending on the combination of the factors the offspring inherits. And Mendel's factors are called genes. The term Mendelian trait is used to describe a characteristic caused by a single gene that sometimes reappears in 1/4 of the offspring. That characteristic can be innocuous, such as freckles or the ability to curl your tongue. But it also can lead to serious illnesses like cystic fibrosis or Tay-Sachs disease. Imagine all that from one man's work with the humble pea.
Okay, so that wraps up the screencast on Mendel's work. And in the following screencast after this, we'll talk about a little bit about the 3 laws that Mendel had come up with. And then also a third screencast that kind of deals with Mendel will be looking at codominance, blood typing, incomplete dominance. Okay, so that's screencast session number 2, dealing with Gregor Mendel and his work. And have a good day.